How does psychedelic intake impact the gut microbiome and the gut-brain axis? A story from a psilocybin mushroom's point of view.
Psilocybin mushrooms, like myself, are commonly known as magic mushrooms because we contain the psychedelic compound psilocybin. The use of these psychedelic mushrooms has a long history dating back to ancient times. For example, indigenous peoples of Canada have used us for spiritual and medicinal purposes for thousands of years, and in the 1950s and 1960s, psychedelics gained popularity in the Western cultures, with many people using us as means of self-exploration and expanding consciousness. If we flash forward to today, we see that humans actively use us as recreational drugs, and research on the potential medicinal benefits of our powers is expanding. In recent years, small-scale studies on psilocybin have elucidated a possible pathway for remediation of anxiety and depressive disorders, but to date, few have investigated its possible synergistic interactions with naturally existing precursors, and none have looked at the impact on the gut microbiota. Similarly, psilocybin remains untested in a rat model, with the few available studies being performed in mice. In this video, I hope to provide a foundation from which future research on the synergistic effects of psilocybin and its respective effects on the gut bacteria can be built upon. I will take you along my journey from consumption to the influence on the brain by interacting with the gut microbiome. So come along with me as I travel through the human body. So where do I begin in my life? My life starts in a forest, being stuck in the ground next to my friends and family waiting to be found. Once I'm found, I could not be happier because I know I'm off to help someone and heal them in some sort of capacity.
The next part of my journey occurs when a human finds me and consumes me. I then start my travels through the human body, starting with the GI tract. Since us psychedelic mushrooms are serotonergic drugs, we love to bind to serotonin receptors. Serotonin is largely produced in the gut. Did you know that 5 of the 7 known serotonin receptors are located here in the gut? One very important serotonin receptor is called 5-HT, which we will discuss in more detail in a second. 90% of the body's supply of serotonin is synthesized in special cells called enterochromaffin cells in the GI tract. Enterochromaffin cells work directly with the gut microbiota, but also they work directly with both efferent and afferent synapses. The 5-HT serotonin receptors are produced by the enterochromaffin cells, and these are our favorite receptors to bind to. You are probably wondering why these are our favorite receptors, and it has to do with the gut microbiome. The gut microbiota are very important as they control about 50% of serotonin that is derived from the gut. It is possible that we and other serotonergic drugs interact with downstream receptors of the host and that gut microbes sense and metabolize serotonin. It is important that we respect the gut microbiota and work with them so that the lasting impacts to mood and behaviors by us psychedelics are mediated in part by gut microbes to create a positive outcome all around. Furthermore, when learning about the 50% of serotonin derived in the gut, there are 2 main bacteria that dominate this regulation, and they are a big deal to us. The next step in our journey also occurs in the gut, specifically the gut-brain axis. As I just mentioned, us psychedelics have to bind serotonin receptors, and 5 of the main receptors are located here in the gut. When something binds to serotonin receptors, they release either an excitatory effect or an inhibitory effect. When we bind to the serotonin receptors, an excitatory response occurs and travels up the gut-brain axis. The gut-brain axis allows for bidirectional communication between the central and the enteric nervous system, linking emotional and cognitive centers of the brain with the peripheral intestines. These communications between the gut and brain can occur by neural, endocrine, immune, and humoral links. The role of the gut-brain axis is to monitor and integrate gut functions, linking the emotional and cognitive centers of the brain to the intestines, and for immune activation. The excitatory response from us binding serotonin receptors mainly travels through the neural and afferent axes up to the brain. Additionally, some hormones are released as some serotonin travels in the endocrine pathway portion of the gut-brain axis. This excitatory response that happens because of us psychedelics binding to the serotonin receptors balances different neurotransmitters in the brain, helping to create better moods and more positive emotions. Some of the neurotransmitters being affected other than serotonin are GABA and dopamine. Serotonin also helps to regulate sleep cycles, appetite, and pain control. These roles that serotonin plays makes it easy to see how us psychedelics could be used to treat different disorders like depression and anxiety.
Based on my brief overview of the relevant literature about serotonin, the gut-brain axis, and classic psychedelics, I will now present a hypothesis about the specific psychosomatic mechanisms of serotonergic psychedelics. I've included a figure so that it is easier for you to follow along with the mechanism that I am describing. Beginning at the bottom of the figure, we can see that serotonergic psychedelics affect gut microbes that produce a temporary increase of 5-HT by their host enterochofman cells. This increased 5-HT production is taken up by the distributed by platelets and may work as a hormone-like regulatory signal which could influence the membrane permeability in the organs, tissues, and brain of the host. This platelet-mediated distribution of the intestinal 5-HT is possible, as 5-HT has a strong lipid membrane affinity, influences membrane structure properties that modify membrane signaling processes, and influences vascular permeability. Additionally, several studies have found that increased plasma 5-HT levels can increase the permeability of endothelial cells as well as the blood-brain barrier. Therefore, this enhanced permeability of the blood-brain barrier could allow for plasma 5-HT to enter the CNS and be distributed, as seen at the top of the figure. As we make our way back down the right side of the figure, we can see that the gut-derived 5-HT, once diffused through the CNS, could modulate excitatory and inhibitory neurotransmission. Additionally, it could create a quick disintegration in the normal neural network by reducing connectivity between the frontal cortex and the lower brain areas and promoting structural and functional neuroplasticity. This would effectively allow patients to access suppressed fear information in their brain and perform an emotional reset. This would also allow patients to process traumatic memories and stressful experiences.
I will be discussing an intriguing area of research concerning the potential therapeutic effects of psilocybin, the compound found in magic mushrooms, on mood disorders and its impact on the gut microbiome. Again, I am psilocybin, here to guide you through this fascinating topic. While the exact mechanism remains hypothetical, current research on other fast-acting anti-anxiety and antidepressant psychedelics like ketamine has shown significant and rapid changes to gut microbe composition. One proposed mechanism is my potential to activate intestinal serotonin receptors, which could affect the expression of genes involved in immune function and inflammation, and alter the relative frequency of certain gut bacteria. However, I'm not currently accepted for medical use, which has hindered advancements in understanding my efficacy and safety for treating mood disorders. Nevertheless, alternations to the gut microbiome suggest that I could positively affect the gut-brain axis. This opens up the possibility for future research to investigate how I might modulate the gut microbiota to treat both dysbiosis and other disorders. Numerous studies have shown my potential in treating various mental health conditions, such as depression and anxiety. My ability to modulate gut-brain axis could provide additional therapeutic benefits beyond my direct impact on the brain. This interplay between the gut and brain is an exciting frontier in mental health research, and I could be a key player in unlocking new treatment strategies. It is essential to recognize the challenges faced by researchers in studying my therapeutic potential due to my current legal status. However, as more research is conducted and the understanding of my benefits expands, the possibility of my being accepted for medical use could increase, paving the way for further advancements in the field. The relationship between me, the gut microbiome, and mental health is an emerging and fascinating area of research. By delving deeper into the mechanisms underlying the gut-brain axis, including my potential to activate intestinal serotonin receptors and exploring my potential to modulate gut microbiota, researchers may uncover new ways to treat mood disorders and improve overall mental well-being. As we learn more about this promising compound, we can look forward to a future where innovative treatments for mental health disorders may become more accessible and effective.
I never imagined that one day I would be such a huge topic of research for humans. But here we are today discussing the therapeutic benefits of psilocybin, the psychoactive compound found in psychedelic mushrooms like myself. Humans have found that psilocybin has the potential to positively impact mental health, mediated by its effects on the gut microbiome. My psychoactive compound can alter the gut microbiome by promoting growth of beneficial bacteria. The gut-brain axis provides a way for me to also positively impact brain function. Clever researchers have noted how I can be used to reduce symptoms of anxiety, depression, addiction, and post-traumatic stress disorder. In a therapeutic setting, it has been shown that my psilocybin can have both anxiolytic and antidepressant effects. I should warn you, it is risky to use psychedelic mushrooms like me straight out of the ground as treatment for mental health. That's why scientists have come up with ways to create my psychoactive compound synthetically. That way professionals can give you the right dose, perfect for you. Trust me, it is much safer and more reliable that way. Despite the potential risks, psychedelics like me can offer hope for individuals struggling with their mental health and provides a new path for promoting overall well-being. I am so excited for the opportunity to further science and lend a hand in the medical community.
Let's jump forward 20 years to see how psychedelic use has developed in the future. The stigma is gone. People finally see me for who I am, which is a happy mushroom that can help the human body in all sorts of ways. I followed the same legalization trajectory of marijuana and became legal in the United States in 2037. Not only that, my powers are still being tested in research with the industry growing by almost 20%. I have no idea what the future holds, but I hope that with further research, I can help many more disorders or illnesses by manipulating the gut microbiota and helping all of mankind.
Thank you for letting me share the story of my species, psilocybin mushrooms. I hope you were able to learn how psychedelic intake impacts both the gut microbiome and the gut-brain axis. Additionally, we covered some very important topics today, including what is known about psychedelics like me, what's known about our relationship with the gut microbiome, how we are being used today in 2023, and finally, what's in store for us in the future. I hope this proves to you that psychedelics are much more than just a recreational drug. Our health benefits could possibly be endless. It's also important to note that psychedelics are non-addictive, therefore, you humans and us psychedelics should team up and work together to help out humanity. Thank you for listening, and please ask any questions you have for me now. I have trained these humans to answer correctly. Thanks!